Hi, welcome to Neuroverse, a podcast hosted by Clara and Carolina, where we discuss all matters from neuroscience to philosophy and beyond.
Today's episode is on understanding the neurobiology of schizophrenia in animal models, and we are joined by Dr. Alice Petty. Alice completed her PhD at the Queensland Brain Institute in Australia, where she developed a new animal model of relevance to schizophrenia, the EDIPS model. She was subsequently awarded an Endeavour Global Leadership Award and recently the Marie Sklodowska-Curie Individual Fellowship, which allowed her to take up a postdoctoral position at Imperial College London, where she continues to work on the neurobiology of schizophrenia and apply the EDIPS model to explore the association between hyperdopaminergia and neuroinflammation in schizophrenia. Thank you so much for joining us today, Alice.
Thank you for having me. We're really excited to be here.
So, to begin today's episode, I wanted to just start with a general question of what are we talking about when we're talking about schizophrenia in a research context, and how can we study such a human-specific disorder in animals?
Yeah, look, it's a great question. I think schizophrenia is, obviously as a psychiatric disorder, the symptom domains are huge and encompass a lot of different issues, different symptoms, including the sort of cognitive symptoms and negative symptoms. The positive symptoms, we call them positive because they're like additions to normal behavior, things like psychosis and hallucinations. Those are, we kind of think they're pretty much exclusively human experiences. And obviously, this is a huge challenge for anyone who wants to look at schizophrenia using an animal model. And so the approach that we have is to kind of, investigate this topic more indirectly. So maybe we can't model the entire complexity of a human disorder like schizophrenia, but what we can do is try and unpick some of that neurobiology. And the part that I'm really interested in and have been focusing on for a long time is this idea of too much dopamine in the striatum. And that is something that we can model in particularly rodents, is the species that I work in, because a lot of those systems are conserved in animals, basically. And that means that we have the capacity to manipulate aspects of dopaminergic neurotransmission. And then what we want to understand is, what does this mean for other parts of the brain? How does this develop? And also, what can we do to sort of ameliorate it, right? To look at potential ways to treat schizophrenia, since the options clinically are pretty inadequate at the moment.
Yeah, so this sort of like reductive approach does make sense in terms of understanding the neurobiological basis.
Yeah.
So you mentioned the involvement of the dopamine system. As far as I know, there are many different neurobiological alterations thought to be involved in schizophrenia, as well as you mentioned, all of these different behavioral phenotypes, like the positive and negative symptoms. So does the changes in dopamine, do they affect the positive or negative symptoms in schizophrenia?
Yeah, this is like a huge question in schizophrenia and still undecided, basically. I guess the current thinking is probably that the dopaminergic hypothesis mainly explains the positive symptoms. So the psychosis, the hallucinations, delusions, that kind of thing. And that's thought because of various neuroimaging studies that have shown a correlation between hyperdopaminergia and some of those symptoms. Also, if you induce hyperdopaminergia with drugs like, for instance, amphetamines, you can kind of exacerbate psychosis symptoms even in otherwise healthy people. So there seems to be a pretty clear link between hyperdopaminergia and the positive symptoms. And it would be really interesting to know, and it's something that we're all very interested in, is whether those also result in the negative and the cognitive phenotypes as well. The other side of the too much dopamine in the striatum is a deficit of dopamine in, for instance, the prefrontal cortex. And that's a region that's really important for a lot of cognitive function. So it seems as though it's not just about the changes in dopamine, but where they're happening that's going to affect the kinds of symptoms that you get from that. And they're all kind of connected. I mean, the brain is not like, you can't think about one area in a vacuum, I suppose. So when you talk about dopaminergia in the striatum, obviously that can contribute to downstream factors in all kinds of other regions and also implications for other neurotransmitters as well.
Yeah. I wanted to ask about, the animal model that you've created or worked on. So I know that it's related to the schizophrenia prodrome. So I thought I would ask firstly, what is the schizophrenia prodrome and what are the benefits of understanding the prodrome?
Yeah, absolutely. So a prodrome is quite a general term, I think, for just a sort of pre-diagnosis phase of a disorder. So you can have a prodrome of many different sorts of disorders. And with schizophrenia, so to reach a diagnosis of schizophrenia, you have to kind of hit a threshold of certain symptoms and certain disruption to normal life is when it will tip over into that actual diagnosis. But before that, you can start to have an onset of symptoms, which might be quite mild. So perhaps you have episodes of psychosis, but maybe they're quite limited, maybe they're quite short in time or, not intense, I suppose. And you might also have the onset of some of the cognitive symptoms that are typical of schizophrenia, issues with all kinds of, memory processing, executive functioning, that sort of thing. And also the negative symptoms, anhedonia, social withdrawal, that kind of stuff. And that can happen from, basically the peak onset time for schizophrenia is early adulthood, so approximately 25 or thereabouts. But these early symptoms can start quite a lot before that, so from 16 onwards, basically. The clinical side of things is not my speciality, but essentially you've got this kind of progression of symptoms. And what's really interesting is that seems to align with a progression of neurobiology as well. So this hyperdopaminergia that we see in people with kind of established schizophrenia also seems to start in that prodromal phase. And it's at kind of an intermediate level between like healthy control people and people with established schizophrenia. And so this is quite interesting because it means that we have the potential to intervene, right? If we had a perfect way of identifying people who were going to develop schizophrenia, it might be possible to intervene before they hit that kind of diagnosis criteria and to realign their neurobiology to a healthy pathway rather than the pathway that leads to like this diagnosis. But of course, in order to do that, we need a good animal model, right, to see that development of hyperdopaminergia and what it means for the developing system, particularly because, of course, the adolescent brain is really like malleable, undergoing a lot of changes in dopamine and in all kinds of other, structural regions and other neurotransmitter systems. So it's a really flexible brain at that point, which means it's quite vulnerable, but also means there's a lot of potential for realigning that trajectory onto a healthy pathway. Yeah.
Yeah, I think this is so important. Like the idea of understanding the neurobiology of so many different neurological disorders is that the neurobiological changes do seem to often occur before any of the behavioral phenotypes. Like you see the same in even Alzheimer's disease or other neurological disorders. So yeah, I definitely think that is where the value of having these animal models lies, is that you can hopefully relate it to the behavior as well as you can, but you can also understand potentially what's happening before that.
Yeah, absolutely. Yeah.
I'm curious to understand a bit better with the onset of schizophrenia so late in comparison to other disorders such as autism, where you have the symptoms quite early on. Yeah. Is there a shift or a cascade or something that does trigger that change?
Yeah, look, it's a really interesting question. I think, again, this is one of the key things that we'd like to understand. Is there a shift and at what point does it happen? And it kind of goes back, I suppose, to the general etiology of schizophrenia, which is still incompletely understood. There's a huge genetic component to schizophrenia. It is a very heritable disorder. But there can also be a lot of environmental factors that influence whether you're going to end up with a diagnosis of schizophrenia. And those environmental factors can be really early in life. They can be prenatal, perinatal factors. Maternal immune activation is one of the classic ones that we model in rodents as well. So, a pregnant mum experiences an infection of some sort, and that can lead to a higher risk of getting schizophrenia in the offspring, but it's certainly not guaranteed. So I think one theory is that you can have a sort of genetic vulnerability and then potentially an environmental sort of insult on top of that will unmask the symptoms of schizophrenia. And that can happen also a bit later in life. So in adolescence, for instance, some types of drug use have been known to flick that switch, I suppose, and stimulate whatever cascade is happening that might lead to schizophrenia. Yeah, I mean, this is one of the complexities, is that the etiology could come from any angle, right? And it has been quite hard to unpick that. And I suppose with the animal models, what we're looking at is where do those environmental and genetic factors kind of converge biologically? And the dopaminergic pathway seems to be one of them. in that all those different, genetic factors, different environmental factors, and, things happening in adolescence all seem to result in this dysfunction of dopamine. So that's one of the reasons it's called also the final common pathway of schizophrenia, suggesting that it's this like convergent point at which schizophrenia may then result.
I may be out of my depth, but could it have anything to do perhaps with like an excitatory and inhibitory balance? Because I know that, for example, when looking at animal models, I don't know if this is true for humans as well, in that initially when they're neonatal, it's all just excitatory, and then only after a certain point it becomes excitatory and inhibitory. And then I guess this also potentially relates a little bit to the dopamine, in that you said that a lot of the focus is dopamine and striatum, and there's the D1 and D2 receptors that act very much in opposition. So is that perhaps the source of an imbalance?
Yeah, absolutely. Yeah, look, the excitatory inhibitory hypothesis, I think, is definitely playing a role in schizophrenia. And it could be playing a role, as you say, at a lot of different developmental time points, right? So, the transition and the balance of those neurotransmitters adjusts from, I guess, prenatal life into adolescence, into adulthood. And you might have a disruption at any of those points, which is going to affect other aspects of the, brain neurochemistry. The aspect of that I'm most familiar with, I think, is about the midbrain. So the midbrain is where the dopamine neurons that project to the striatum are located. And the midbrain typically gets a lot of inhibitory influence from a lot of GABAergic input from local regions like the reticulata, which is also in the midbrain. And then it also gets excitatory influence from glutamatergic input from the cortex, for instance. And it seems as though in schizophrenia, what might be happening is that you're getting a dysfunction of that balance, which means that the brakes that are put onto those midbrain neurons come off a bit. You get elevated firing of those midbrain neurons, and that's causing or contributing at least to this excess of dopamine in the striatum. So that's one small example, I suppose, of that excitatory inhibitory balance being kind of dysfunctional, at least hypothetically, I think.
Yeah, I think that's a complex aspect to translate from animals to humans, because I know that excitatory inhibitory balance is implicated in basically all neurodevelopmental disorders, which include schizophrenia, but also autism, epilepsy. And GABA, the main inhibitory neurotransmitter, is known to be excitatory very early on in development. But that has never been properly shown in humans because it's so difficult to show. It's known that it's implicated, but I think that will be a major challenge to investigate in humans.
Yeah. I mean, it raises an interesting point. Like, we kind of only know about the things that we can measure in a way. And it just so happens that we can measure dopamine quite well because we have technology that can do it. For instance, a lot of the clinical data that I work on uses PET, positron emission tomography. And for that to work, you have to have a radioligand, just an aspect of neurobiology that's labeled with a radioactive marker so that you can see it in the brain. And those are more easily made for some ligands than for others. And that means that we're naturally going to have a bulk of information about maybe one neurotransmitter system rather than the other. So I'm not sure what the situation is with GABA, but maybe there's just a lot harder to look at for reasons that aren't to do with the neurotransmitter itself, just that technology hasn't kind of caught up with where we'd like it to be, I think.
Yeah, that's a really good point.
So going back to the animal model, could you tell us a little bit more about the process and specifically what it is about the animal model you developed and how it contributes to the field of schizophrenia?
Yeah, so look, this is one I developed in my PhD. So a lot of it was, when you start a PhD, you're kind of at the whim of your PhD supervisor, I'm sure you can imagine. And he had this idea of using this genetic construct, which was originally designed to treat Parkinson's. So Parkinson's deficit, too little dopamine in the nigra, in the midbrain. And he wanted to kind of repurpose it, I suppose, to elevate dopamine in otherwise healthy animals to try and replicate this schizophrenia neurobiology. So yeah, basically it's a construct that codes for two enzymes that are key for synthesizing dopamine, which you can imagine could be extremely useful if you had Parkinson's. And then putting it into otherwise healthy animals, we see that they show some of these behavioral outcomes of hyperdopaminergia that we were talking about before, like elevated hyperlocomotion, for instance, We did also have a look at some of the negative and cognitive phenotypes just to see if they were affected as well. It seems like they weren't, but that's still something that we're keen to follow up on because, as we said, it's really interesting, would be really interesting to know. Yeah, that's, I guess, the origins of the model. And then we also, the prodromal aspect of it is that we inject this construct into quite young animals, so 5-week-old animals rather than adults. So 5-week-old rats is kind of a juvenile time point, sort of peri-adolescent, I suppose. And we did that because we were really interested in mapping that like progression of dopaminergic dysfunction over that really key developmental period, basically. And we did find that the behavioral phenotypes come on progressively, just as you would see that elevation of symptom intensity in the prodrome to schizophrenia as well. And I suppose the other important aspect of it, as well as that temporal specificity, was that with a genetic construct which is packaged into like a viral capsid, which means you can deliver it to a specific region in the brain. And we delivered it into the nigra so that it would project up to the striatum, and specifically the dorsal striatum, which is a region that's most established as having this dopaminergic dysfunction. So, I mean, it's, no model is perfect, and it's even now undergoing, there's EDIPS 2.0, there's probably going to be EDIPS 3 and 4. Like, it's a really iterative process, especially because the, clinical findings are advancing as well as we speak, really. So a big part of animal models, I think, is trying to optimize them really and try and unpick different aspects of the neurobiology that could be really interesting. And one of the projects that I am working on at the moment is undertaking PET scans, preclinical PET scans in these animals, because PET is a technique that's been used in humans a lot and has revealed all these dopaminergic abnormalities, basically. And it would be really helpful for us if we could use exactly the same technique to find that same dopaminergic dysfunction in this preclinical model. It would be so useful in terms of like translatability, trialing new therapeutics, that kind of thing. So yeah, I mean, there's a lot of angles that we could be following up on, I think, with the EDIPS model.
Reading about the model, it seems particularly appealing to me that it seems to replicate the changes that occur in schizophrenia over time, like how the changes in other neurotransmitter systems, like the glutamatergic system and the serotonergic system, are also replicated, even though you're only altering the dopamine, right?
Absolutely.
Yeah. But I was wondering, because you mentioned earlier that the dopaminergic pathway is seen as the final common pathway. So how does altering that lead to these other changes, which are thought to lead to that?
Yeah. I mean, when I say, dopamine is the final common pathway, half of schizophrenia researchers probably aren't focused on dopamine. And there are many other hypotheses that have a lot of other literature behind them as well. I think, the exact way in which altering dopamine in the striatum might affect glutamate is still unclear. I mean, the striatum, it feels like a real hub, I suppose, of the brain. It's a key part of this basal ganglia circuit, which projects up to the striatum, forms this kind of feedback loop through a lot of other different brain regions in quite a polysynaptic way. So there's likely to be all kinds of complicated, I guess, downstream effects of manipulating striatal output, really. So I suppose, the upshot is I'm not sure yet. I didn't have a chance to follow that up a lot in my PhD. And it would be really interesting because a lot of the glutamatergic models do seem to replicate more the negative and cognitive symptoms of schizophrenia. So it'd be really interesting if your original manipulation is just dopaminergic, but then you're getting all these downstream effects in these kind of companion neurotransmitters. Does that mean that the dopamine dysfunction is where it starts? Or is this just one mechanism of getting to those glutamatergic abnormalities? Like, do they work in parallel? Could you have like linked dysfunction, I suppose? Yeah, I mean, there's just still a lot that we don't understand fully.
And then I wanted to ask, related to what we were speaking about earlier in terms of environmental effects, have you used this model at all to look at the interactions with environmental effects, like inflammation, for example?
So, some work that I was doing in Sydney at Neura was looking at the EDIPS model and this idea of neuroinflammation. And we did some RNA-seq on midbrain tissue from these animals. And what we found is that a lot of inflammatory processes seem to be upregulated. So it seems as though increasing dopamine in this midbrain region affected local inflammation. And the reason we were really interested in that was that this group with Professor Cindy Shannon-Weickert has looked at the midbrain as a locus for inflammation in postmortem tissue, so from people with schizophrenia and healthy controls. And she finds that there's a subgroup of people with schizophrenia who show elevations in inflammation in this region. And so we were really interested in seeing whether EDIPS would happen to model that and all the kind of cascading biology that's associated with upregulated inflammation. And it seems like there is. I mean, it was very, very pilot work, but it's something I'd be really interested in following up, because I think it's a really interesting hypothesis of schizophrenia, I suppose, that a big chunk of it is this kind of immune response that's gone wrong. And whether that happens before or after dopaminergic dysfunction would be really interesting to know, like which one comes first, or do they happen simultaneously, that kind of thing.
Yeah, because I've only read about it in terms of like maternal immune activation leading to the deficit. So I assumed that if you were looking at inflammation, it would be as a cause rather than a consequence. So that's really interesting.
Yeah, it's something we'd love to follow up, because it really raises questions about what comes first, I guess.
Mm-hmm. Yeah.
So having taken these approaches by altering the available dopamine, do you then also do behavioral experiments? Because as we've mentioned, schizophrenia is a very human-specific disorder. So how can we then relate the animal behavior to the human behavior?
Yeah, look, another great question, and also a huge limitation of animal models. We have to accept that, we're never going to be able to mimic psychosis. So the sort of behavioral measures that we have are pretty indirect at the moment, or at least the commonly used ones. So a big one is just hyperlocomotion. So basically, you put an animal in a box, you give it amphetamine, and you look at it run around, and more dopamine means more running around. And that's kind of a proxy readout for hyperdopaminergia, which is kind of a proxy for the kind of neurobiology potentially underlying psychosis. But that is, as I say, a really blunt tool that we have to look at that sort of thing. There's a lot more nuanced work, I think, going on now with more involved behavioral tasks, where animals can, for instance, be asked whether they see a signal or not, like a light signal or an auditory signal. And you can look at how confident they are in that signal, and whether they're sort of seeing a false signal where we wouldn't expect that. And that could be connected to some aspects of, for instance, a hallucination. But that's still quite early days, and it's certainly not the commonly used techniques. So look, it's a huge challenge, I think, because there's a lot of overlap in behavior, for instance, with rodents. But for these positive symptoms, it is really hard to model them well. And that is an issue with the field currently, and an issue in terms of translatability of the results that we find. I mean, if we want to be targeting a human disorder, we really need to have outcomes that, match what's going on in people. And that's a really tricky problem to solve, I think.
The example of the study that you mentioned about, I guess, putting visual cues and seeing whether or not the mice respond to them, I think that's really interesting, because something we haven't covered yet, and I'm sure you can explain it in a much better way than I can. But what I understand in terms of the internal model of schizophrenia is that there's quite a strong top-down projections in the brain that rely a lot on contextual information, and external information, so bottom-up signals, either don't really have as much of an impact or don't really get registered. I'm not quite sure exactly how to explain it, but from my understanding, there's a much stronger top-down component. And so, what you mentioned about the behavioral experiments of visual stimuli make a lot of sense. Would you be able to comment on that?
Yeah, I'm not super familiar with the kind of model idea, I suppose, of that top-down, bottom-up processing. But there's certainly evidence that sensory input is being processed abnormally in schizophrenia. And exactly where that's happening in the brain is not quite clear, and probably depends on the kind of input that you're getting. We had an interesting paper in a journal club recently about auditory hallucinations, kind of tapping into this idea of auditory hallucinations as internal monologue that gets misattributed to something external to a person, and giving this impression of it being, a voice from outside of oneself. In terms of animal models, another classic behavioral test that we do is called prepulse inhibition. So prepulse inhibition is kind of looking at this idea of sensory gating, I suppose. So in animals and in humans, actually, if you provide a loud tone, but before that you do a little prepulse tone, That pre-pulse means that we respond less to that stimulus. And this is thought to be a way of kind of gating the inputs that are coming into the brain, basically. And people with schizophrenia show a deficit in this pre-pulse inhibition. And it's something that we find in a lot of animal models as well. Yeah, whether that's a generalizable kind of deficit, I suppose, in how inputs are processed is not something I know very well. I know it encompasses a lot of different regions in the brain and a lot of different neurotransmitter systems as well, because, you can imagine that different regions would be lit up depending on the kind of sensory input that you're getting, whether it's auditory, visual, or otherwise. Yeah, really interesting idea, the idea of, how is the brain integrating this information that's coming in, and what kind of dysfunction is going on in the kind of modulatory regions, I suppose, which affects how people perceive external stimuli. Again, not really my field, but I think there's a lot of push to do more nuanced testing in animals using, for instance, little touch screens where they can choose different images, lots of cognitive testing with light signals coming in, visual stimuli coming in, which probably tap into that kind of idea of sensory processing a bit more than the basic, hyperlocomotion, for instance.
Mm-hmm. Yeah, really interesting.
Yeah, I guess it's this idea of the predictive coding theory that we've talked about in a lot of our previous episodes on Neuroverse.
Yeah.
Mm-hmm.
Yeah.
So alongside animal models, we're often also very limited by the technologies that are available to us. Are there any emerging technologies or like AI or even other animal models that you think are really beneficial to studying schizophrenia and are going to make an impact in the near future?
Yeah, that's a really interesting question. In terms of AI, who knows? I mean, it has enormous potential, right? But I think it could have a huge potential for integrating a lot of data, especially some of the imaging data coming out from clinical studies. I think will really benefit from looking at, for instance, correlations between different regions, whether those are related to different, symptom domains, that kind of thing. And in terms of other animal models, I mean, a lot of the work that we do is on kind of tried and true models. I'm doing a lot of work in the ketamine model of schizophrenia at the moment. So we give ketamine for 5 days and we see that those animals show hyperdopaminergia. And ketamine is a NMDA receptor antagonist, so it's tapping into this glutamate hypothesis rather than the dopaminergic hypothesis, but it seems to be connected to this hyperdopaminergia idea. I don't know if I'm aware of really fresh animal models coming out. I mean, there's so many that are generated at all times related to lots of aspects of schizophrenia, a lot of genetic manipulations going on. There's a lot of potential with, some of the new technologies about gene editing, that kind of thing, and technology around, for instance, chemogenetic activation of quite discrete anatomical regions. And they can all kind of be mixed and matched, I think, into hugely powerful animal models. And I'm really excited to see what's coming up in the field, basically. Yeah.
Sounds exciting.
On that note, though, you did mention about the use of AI to look at big data and maybe associate the genetic differences with different symptomatic domains and that sort of analysis, which I know is becoming like increasingly done as we gather larger data sets. Again, my knowledge of this comes from the world of autism, but I'm sure it's similar in schizophrenia. So do you think that because of this sort of research, which can be done using human data, there is going to be a reduced need for animal models?
Certainly, I hope not. Look, it is possible. I think they can be quite complementary, I think, the clinical work and the animal models, especially with schizophrenia, although it is, has this huge heritable component, it's really polygenic, same as autism, I'm sure. There's no one risk factor that's likely to contribute to disease phenotype. And that means it's really hard to unpick which genes are going to be most, involved, I suppose. And you can generate these polygenic risk scores, I think, something that I haven't done actually, which kind of weigh up all the different genes and kind of give you this risk potential for developing schizophrenia. A lot of animal models have looked at manipulating those genes that are risk factors for schizophrenia and seeing what those genes contribute in terms of the phenotypes that we see in those animal models. And that's one way of trying to figure out, what do these genes actually mean in terms of symptomatic outputs. And that's something that's really hard to do in a clinical setting.
Yeah.
Because, you can't manipulate the genes of a person, and certainly not a developing person, in the same way that you have the potential to, especially with these, as I mentioned before, genetic, gene editing potential, knocking down, silencing specific genes, even in specific regions, for instance. So I think animal models will hopefully still have some advantages over clinical work, but I think they'll definitely move along in parallel, would be my thinking. So as new evidence comes out from, for instance, big data analysis, I think that's likely to inform where new animal models are going to focus, because it's it's a quickly moving field. Schizophrenia research has been around for many decades, but it's with these new technology advances, it's being probed in a very different way, I think, to how it used to be. And probably more nuanced, and probably revealing more subgroups. There's an idea at the moment that schizophrenia, although the diagnosis is just one clump, in fact, it probably reflects a whole lot of different kind of sub-disorders, really, which all perhaps have similar symptomatic expression, but the neurobiology might be really different. And I think that's somewhere that the field is heading at the moment, and somewhere that big data and AI might be really useful.
Absolutely.
Yeah, really well said. Thank you so much for joining us today, Alice, and sharing all of your knowledge. We've covered a lot, and it's been really insightful.
I had a great time. Thank you for having me.
Thank you for joining us. Yes, it's been fascinating to have an insight into the development of animal models.
And you certainly have proved that they are here to stay, at least in the foreseeable future, and they are definitely useful for understanding these disorders. Thank you so much, everyone, for listening. If you enjoyed today's episode, do share the podcast with your friends and family, and be sure to follow us on social media on Twitter and Instagram and LinkedIn.
And don't forget to check out our website for links of all the resources for our episodes. And thank you for all your support.